For the next oral presentation is identification of receptor binding sites and conserved regions among published enterovirus serotypes using bioinformatics and EGRET neural network for multivalent vaccine development, presented by Win Warasap Paisan team.
Hello. Our research was conducted by Winwar Sapaisan, Porichanasan, and Tawin Supachok Satapon. Today, we will present our findings on the identifications of receptor binding sites and conserved regions among enterovirus serotypes using bioinformatics and EGRET neural network for multivalent vaccine development. Enterovirus is in the families of Piconaviridae. They cause broad range of diseases, including poliomyelitis, hand-foot-mouth disease, and viral meningitis, and also AFP, acute phase paralysis, with more than 300 serotypes identified. Their high genetic diversity creates a major barrier in current universal vaccine design. Currently, only inactivated EVA71 vaccines are widely available. They provide incomplete protections, covering roughly 26% of circulating strains in Asia. As a result, millions of infections and deaths every year. Laboratory-based structural and antigenic analysis remain slow and costly. These limitations highlight the need for computational process that is capable of identifying rapid conserved sites and receptor binding sites across diverse serotypes. Our research has two main purposes. First, identifying conserved regions and receptor binding sites across multiple enterovirus serotypes to pinpoint optimal vaccine targeting sites for multivalent vaccine development. Second, to develop a software to pinpoint optimal vaccine targeting sites for multivalent vaccine development.
The enterovirus capsid is formed from repeating promoters, each composed of VP1 to VP4 proteins encoded in P1 region. And for the methodology, RNA sequences from 80 enterovirus species were retrieved from GenBank. Sequences were translated and structurally modeled using the AlphaFold tree. Next, multiple sequence alignment was performed and conservativeness was quantified by percent identity. This allowed us to locate the most stable areas across different virus cell types. Next, interaction propensity with the host receptor SCARP2 was predicted using an AI neural network, EGRET, where higher scores reflect stronger likelihood of viral host protein interaction. Last but not least, conserved residues and high propensity sites were mapped onto a 3D structures using PyMo. And lastly, two clinically significant groups were analyzed separately, first one being the HFMD infectious strain, and the next one is AFP endemic strain.
From our analysis of 80 enterovirus species, we have identified two significant sites. The first being a region adjacent to the VP1 canyon, and the second being the VP3 bulging loop. These two sites show correlation peaks for conservation across multiple species and interaction rate with receptor binding site, meaning that these two sites are suitable for vaccine development. Next, for infectious strain analysis, we have also identified two significant sites. The first being a small point adjacent to the VP1 canyon, and the second being a broad region beneath the VP3. For AFP endemic strain analysis, we have identified three significant sites. The first being a region adjacent to the VP1 canyon, second being a VP3 bulging loop, and the third being a broad region under the VP3. We conclude that the VP1 region adjacent to the canyon and the VP3 bulging loop to be correlation peaks for conservation and has high interactivity rate for receptor, meaning that the two sites is suitable for broad vaccine development, aiming to create cross-protectiveness against wide range of enterovirus. In contrast, we have identified pre-correlation for AFP endemic strain, signifying that it is possible to create a targeted vaccine that specifically targets and prevents the infection of AFP endemic strain. Slight overlap is suspect on the yellow and pink circle on infectious strain and AFP strain, signifying potential cross-protection development of multivalent vaccine. And lastly, in the red arrow here, it is the broad region under the VP3. Though it has high correlation peak, it is unsuitable for multivalent vaccine development as it is not surface exposed. From the pipeline of our research, we have developed web vaccine. It is a software that aims to analyze multiple virus structure to identify the vaccine targeting site. Our software works by combining two data. The first being a conservativeness across multiple virus structure, and the second being interactive rate of receptor binding site. The software will combine these two data together to calculate the SRC value. This value signifies the suitability for vaccine development. Then the software will show the SRC value onto a protein structure. Now, let us show you how our software works. First, we will upload the virus DNA sequence or genome sequence onto the software. Then, after we have set our software up and hit analysis, our software will then perform computations. Then, in just a few seconds, they will project the SRC value onto a protein structure. And the software is currently processing. In just a few seconds, they will project the value onto a protein structure. And yes, there it is. The researcher will be able to use this data for further analysis or further vaccine development.
Vaccines was reviewed with input from Microbiology Department, Faculty of Medicine in Chiang Mai University, and achieved 94.26% accuracy when compared to licensed software. Compared to the other software, vaccine is the first software to be able to find the conserved regions and receptor binding sites, and then integrate them into a unified vaccine prominent site, which is important for multivalent vaccine development. The software will be available for $100 per year, enabling researchers for rapid identifications and university research. And thank you for listening. We would be glad to
show you our live demo again, or we will be free to ask any question because we are glad to answer them.
Could your software apply to another family of the virus?
Yes, as long as we have the genome sequence of the virus, and the genome sequence is section for the capsid or the envelope of the virus. As long as we have the sequence, we can use our software to analyze it.
But in your methodology, you rely on the enterovirus only for feeding your model to
yes.
train, right?
The eGrid neural network is AI model that is trained from another researcher, and it is being trained on multiple virus, and we just use the eGrid neural network to analyze enterovirus. But for the web vaccine software, it can be used on all other virus species, as long as we have the sequence for it.
Could you explain a little bit further, because I a little bit confused, because your software, the web vaccine software, have been built upon the information of enterovirus, right? And my question is that if we want to look at another viruses, so we need to develop a new software or not? Or we need to have another kind of sequencing database based on those kind of new viruses to get another prediction?
Yes. For the research part, we use this pipeline. We perform multiple sequence using BLAST software. And in web vaccine, the BLAST software is also part of the calculation. So BLAST software can be used for other virus as well, as it used to find the conserved region. And as for the receptor binding site identification, the eGrid neural network model is trained on all other types of protein interaction, including the capsid and receptor binding site of all other virus. So we don't need to develop a new software. We can use the eGrid neural network model and BLAST software to find the necessary information to determine vaccine targeting site.
So if I want to look at the new type of virus, I can search by your software, but it will use the data from the eGrid AI. Okay, if you have tests about the other virus that have proved the vaccine.
We are currently working on rabies or Lissa virus and rotavirus, but currently we haven't been able to get the result out because it is still work in progress.
what is the challenge part to get that result?
I think the most challenging part is the data retrieval, as we need to find the genome that is complete and have label on VP1, VP2, VP3, VP4 for the enterovirus. It has many sequences on the NCBI database, but it is difficult to select a few of them that has the complete data.
Yeah, that still be a challenging question that we still keep asking. Thank
Thank
you.
you.